All right, everybody, please make sure that you have your fill in the blank notes out, and we're going to go ahead and talk about multiple alleles today. Just a reminder, I hope you're having a good day or a good night, and we'll get through this, and then you can go about your day. So today we want to make sure that you can define multiple alleles and complete Punnett squares that are using multiple allele traits. Also, ABO blood types exhibit multiple alleles, so you want to know about those and know about blood transfusions. So we're going to talk about the first three in the notes, and then when you come back to class, we'll talk about blood transfusions. So multiple alleles. When one gene has more than two alleles, it is said to be multiple allele inheritance. This results in more than two phenotypes for the gene. So previously we've been talking about if you had like a big H and a little h, you could have big H, little h, and little h, little h. So now what's going to happen is you could have big H, a little h, and then a little h prime, or we could say a big H prime, but we have more than two alleles for one gene. One trait has multiple alleles. So there are two examples of multiple allele inheritance we're going to go over. The first one is rabbit coat color, and then we'll talk about human blood types. So rabbit coat color is determined by multiple allele inheritance. So just by looking at this diagram, I want to answer a few questions. The first one is, what is the gene or trait in this example? So it should be hair or fur color, whichever one you wanted to write. The second question is, how many different alleles are present? So remember, alleles are the individual letters. So you have a big C, you have a little c, and then we have CCH, and then we also have CH. So you should have 4 alleles, a total of 4. Which allele is dominant to all the others? Okay, so if I look at all of the genotypes, which allele, when present, makes it a definite fur color? So this would be big C. The reason for that is because if you look up here at all of these genotypes, the big C is present. So what that means is that every time a big C is present, it's going to be producing a protein that makes the rabbit a dark gray color. Which allele is recessive to all the others? So again, which allele is going to show up, or which allele is going to only be that rabbit color if it's the only thing present? And that should be your little c. So if you look up here, little c is the only way a rabbit could be albino. If there's another allele present that's not a little c, it won't be albino. So the only way that you're going to have an albino rabbit again is to be little c, little c. So that's why it's recessive to all the others. Because if you have anything other than the little c, it's not going to be albino. Okay. So let's go ahead and do a sample problem. So we want to cross a chinchilla rabbit with an albino rabbit. So the first thing we have to do is label our Punnett square. So an albino rabbit is little c, and what was your chinchilla rabbit? And go ahead and fill in the blanks. So here's your albino rabbit, and here's your chinchilla rabbit. And then you write your alleles in the boxes. So now you have the 4 genotypes. And if you notice, the 4 genotypes are all the same. So then we want to ask, well, what are the phenotypes then? Well, they all have the same phenotype. So the CCH is dominant to the C. So if you have a CCH, what is going to be present? So they should be all light gray rabbits is what you should have for the phenotypes. If you were going to write that in a phenotypic ratio, you would have 4 light gray rabbits to 0. All right, so now we get to be a little bit more, what I find a little bit more interesting is human blood types. So when somebody says they have A blood or B blood, that's determined by multiple allele inheritance. So what it is if you have A blood, you have a protein on your cell, and it identifies it as A blood. So that protein on your cell is called an antigen. So if you're A blood, you have the A antigen. If you're B blood, you have a B antigen. If you're AB, you have both proteins, A and B, to make you an AB blood type. Okay. So remember we talked about the functions of cell membranes and the functions of proteins, right? And so the cell membrane, if there's a protein on it that helps identify it, okay, that's a type of protein that can help with cell membrane function. So an O blood type then doesn't have any or either of the proteins. It doesn't have the A protein or the B protein on its cell membrane. So by looking at the diagram, why can't an A blood type donate to B? So in other words, what would happen if A donated to B? What would the problem be? Well, here's the thing. A person with A blood produces B antibodies. So a person with B blood produces A antibodies. The antibodies are only going to be present if it senses an invader. So I kind of look at this like a wanted poster where the body knows who to look for that's a bad guy, so to speak. So if you're B blood, your body is on the outlook for these bad guys that look like that A antibody with that shape. So if an A blood cell is donated to somebody that has B blood, the B blood will start producing antibodies, and then these antibodies will bind to the protein on the A blood cell, and that ends up causing blood clumping and or blood clotting, sorry, and cause death. So when they started doing blood transfusions and some of them worked and some of them didn't, it was because sometimes they would donate to somebody who was compatible with their blood type, and sometimes they would donate to somebody who wasn't compatible with their blood type. So let's do another example. Could A blood, I'm going to use a different color, could A blood donate to AB? So what you want to notice is that the A blood has this pattern here, and AB blood doesn't produce any antibodies. There are no antibodies produced here. And you can think about that because the AB blood type has these proteins. So it's not afraid of them because it already has them. It's familiar with them. It knows what it looks like. So when A blood donates to somebody that has AB blood, it would be okay, and it would be a successful blood transfusion because AB doesn't produce any antibodies. So they're the universal receiver. All right, so thinking about this, can you figure out who the universal donor would be? So your universal donor is going to be your O blood, universal donor, because your universal donor O blood lacks proteins on the cell membrane. So when it donates to anybody, even if they produce antibodies, the B blood produces the A antibody, the A blood produces the B antibody. Well, it's not going to produce those antibodies because A blood doesn't have any of those proteins. So it's not going to trigger the immune system to produce those antibodies in the first place. So like I said, we'll talk more about this in class, but there's just like a little brief introduction on how that works. So when we talk about blood types, there are 4 different types and there's 3 different alleles. Scientists call them I, A, IB, and little I. So the first 2 alleles are dominant to I. So what that means is that you'll have I, A for A blood, or you could have I, A, little I. And that's going to be for A blood. It also says they're co-dominant to each other. So if you look at the AB blood type, notice they have both the A and B protein on their blood cell membrane. So that would be IA, IB. And remember in class I've talked about how if you have a trait, a lot of times you're producing, and a dominant trait in particular, you're producing a protein. So if you have IA, you're producing the protein or the A antigen on that cell membrane. So C, before we move on, if you could figure out what would be B and what would be O. All right, so hopefully you determined that for the B blood type, you would be IB or IB little I, and the O blood type would be little I little I. All right, so let's go ahead and do some practice problems here. Show a Punnett square of the possible offspring between a person homozygous for A blood and a person heterozygous for B blood. So homozygous for A blood would be I, A. What about with B blood? Heterozygous for B blood, what would it be? I, B, little I. So we look at our Punnett square. And again, if we were doing our genotypic ratio, you'd have 2 AIBs to 2 IA little i's. Or our phenotypic ratio would be 2 people with AB blood to 2 people with A blood. And that, of course, reduces to 1 to 1. The next sample problem, show a Punnett square of the possible offspring between a person homozygous for B blood and a person with AB blood. So what would the genotype be for a person with B blood? IB, because remember it's homozygous, so homozygous is the same. AB blood is co-dominant. Both of those proteins show up on the cell membrane. You plug that into your Punnett square, and you should get something like this. All right, last but not least, I know that sometimes you've heard people say, well, I have O positive blood or I have O negative blood. So I just want to talk about what that means. That's called the rhesus factor. So that's an entirely different gene, and it's a gene that is determined by complete dominance. So that's what we started talking out with at the beginning of the chapter, meaning if you have the positive dominant allele, or Rh positive, like I've written here, so Rh positive, that means that you have the dominant trait. You're going to produce a protein on the cell membrane that makes you have that positive protein. If you have 2 negative alleles, 2 Rh minus Rh minuses, that means that you won't have that protein on your cell membrane. It's called the rhesus factor because it was discovered in rhesus monkeys. So this is a cute picture of the rhesus monkeys here. So that's where that protein was discovered. And then what I've drawn is an example of a Punnett square between a parent that was homozygous, so plus minus, for blood type. So just off top of your head, is this person exhibit positive or negative blood type? What's their phenotype? So the phenotype for this person would be positive blood type. So they might be O positive or AB positive. What about this person here that's minus minus? What would they be? They would have negative blood. So it's possible for 2 parents to have positive blood and then have offspring that would have negative blood if both parents were heterozygous. I know that was a long section of notes. Thank you for sticking with me. When you come back to class, we're going to go over some examples with this, and bring your questions, and have a great day. Thank you.